Hi friends, welcome to the front row. Today our topic of discussion is immunoglobulins. They form one of the most important components of our immune system, and by definition these constitute a heterogeneous family of serum proteins which either function as antibodies themselves or are chemically related to antibodies. So by this what we can understand is, though immunoglobulins and antibodies are used as synonyms most of the times, they both are not one and the same. If immunoglobulins is this, then antibodies are a part of it. 20% of the total serum proteins are these immunoglobulins, and they are glycoproteins, by which I mean they are formed of carbohydrates and proteins. Carbohydrates, glyco means polysaccharides, and proteins in the ratio of 4 to 18% is the polysaccharides, that is the carbohydrate part, and 82 to 96% is the protein part. They help to opsonize and neutralize the antigens. Opsonize means they help the body to recognize antigens as foreign particles and further act upon it. And neutralize is they see to it that the antigens do not cause any harm to the host by blocking its receptors and other mechanisms which we'll be learning later. And they help in complement fixation. These are secreted by beta lymphocytes or B cells, and each B cell secretes a specific immunoglobulin which is also antigen specific. If there is an antigen A, then only a particular immunoglobulin can bind to it. And also this particular immunoglobulin is produced by a single B cell. This B cell will not be producing any other type of immunoglobulins. This B cell produces a particular immunoglobulin which is particular to an antigen. So these are highly specific. These immunoglobulins occurs in two forms, that is a soluble form which is freely circulating in the blood serum, and the second one is a membrane bound form which is attached to the surface of B cell, and these membrane bound forms are referred to as BCR, which means B cell receptors. These immunoglobulins that are found on the surface of the B cell act as the receptors to the B cell, and these are not soluble. Classification based on the amino acid sequence in the C region of heavy chain. When we'll be learning about the structure of the immunoglobulin, we will be seeing how an immunoglobulin is made of four chains, of which two are heavy chains. These are heavy chains, and these two small ones are the light chains. And each heavy chain and light chain are, are comprised of a constant region and a variable region. These form the constant region, whereas these form the variable region. So based on this constant, the amino acid sequence in this constant region, the heavy chain, these two chains, these are classified into five types, that is IgG, IgA, IgM, IgE and IgD. If anybody is finding it difficult to remember the alphabets that are associated with these anti-immunoglobulins, you can remember it as game D, which means it's a game of diseases, or how whatever you want to term it as. So this way you won't forget which are the letters that are associated with these immunoglobulins. Why these are named so is because these immunoglobulins contains gamma chain, this has alpha, this has mu, this has epsilon, and this has delta chain. So based on these, they are classified into IgG, IgA, IgM, IgE and IgD. Also, based on the amino acid sequence of the C region of the light chain, these two are the light chains, and in this, the region that is below is the constant region, which do not change with different immunoglobulins. And depending upon the amino acid sequence in this region, they are classified as kappa light chain immunoglobulins and lambda light chain immunoglobulins. The general properties of these immunoglobulins, firstly, amount. The IgG forms a major bulk of the immunoglobulins present in our body, that is about 70 to 80%, whereas IgE forms only about 0.004% in our body. And their normal serum level, IgG forms about 1200 milligram percentage, whereas IgA is about 200 milligram percentage, IgM is about 120 milligram percentage, IgD is about 3 milligram percentage, whereas IgE is only about 10 to 70 microgram percentage. This is in about thousand milligrams, whereas IgA and IgM are in hundreds, and IgD is in single digit, that's only 3 milligram, whereas IgE is not even in milligram but is in microgram percentage. This is how you should know the lifespan of immunoglobulins. They are measured in half-life, that is in days, and IgG and IgA do not have a particular lifespan. IgM has about 5 days of half-life, and IgD has about 2 to 8 days, IgE has about 1 to 6 days. Placental transfer, which means from mother to fetus, and the only immunoglobulin that is capable of passing through this placenta is this IgG. So this IgG is the most important immunoglobulin for a fetus. The antibacterial license, that is the property of an immunoglobulin to act against the bacteria. The IgG are a potent antibacterial bactericidal, whereas IgA has some properties, whereas IgM is a very potent antibacterial immunoglobulin. The role of IgD and IgE is unsure. Antiviral properties, IgG is antiviral to certain extent, whereas IgA is the most important immunoglobulin in acting against viral infections. IgM is also active against virus, whereas the roles of IgD and IgE are unsure.
Okay friends, let's learn about the structure of immunoglobulin in part 2. Thanks for watching. Please do like, share and subscribe for more updates. Thank you.